This is the Anthrobiology Podcast, hosted by me, Gabby LaPera. Every 2 weeks, I interview a biological anthropologist about what makes us, us. Hey there, everyone. You're in for another delightful episode of the Anthrobiology Podcast. This week, I interviewed Dr. Janna Andronowski, who is an expert in bone histology. I'm not going to say any more than that because I think the podcast is actually pretty self-explanatory this week. The only two things that I wanted to bring up is a way to remember the difference between osteoblast and osteoclast for all those folks out there taking intro to osteology for the first time. Osteoblast, this is a cell that lays down bone. Think of it as they're having a blast laying it down on the dance floor / the surface of the bone. And an osteoclast breaks down bone. That's the same as an iconoclast, someone who breaks icons. The other thing that I wanted to mention is that if you're really interested in this idea of modeling and remodeling, then you should get familiar with the Utah paradigm. Don't worry, I will post an article about that on the website. I hope everyone enjoys. And as usual, if you have any questions, any commentary, or if you have a cannibal expert for me, feel free to email me at gabby.lapera@anthrobiology.com. That's GABY.LAPERA@anthrobiology.com. Have a wonderful listen. Hello, everyone. Welcome to another episode of the Anthrobiology Podcast. Today, joining me via Zoom from the exotic land of Canada, I have Dr. Janna Andronowski, who is at Memorial University in Newfoundland. Did I say that right?
Newfoundland, yeah.
I tried so hard. I asked her before we started and I still messed it up. But that's okay.
You can call it MUN, Memorial University of Newfoundland. Everyone just calls it MUN.
Oh, MUN. Oh, I love that. Okay, yes. She's from MUN. And I'm really excited to have her here today because I feel like on the show in the past, we've talked a lot about bones and things that you can learn from them, but we actually haven't really delved into the biology of bones. And you actually study that. Can you tell the listeners a little bit more about what your areas of interest are in terms of research?
Absolutely. And thanks, Gabby, for hosting me today. I look forward to talking to you about my work. So generally, my lab investigates skeletal biology from a microscopic and 3D imaging perspective and applications to human health, human anatomy, as well as forensic anthropology. And I have a very multidisciplinary background. I'm an anatomist, an anthropologist, a biologist. And so my research goals are sort of centered at the interface of bone biology, forensic science, and high resolution imaging, particularly synchrotron imaging. And I'll come back to that word a little bit later.
Good.
But a particular focus of my lab is to further understand the dynamic relationship between human bone tissue and different physiological and nutritional stressors, such as hormones, infection, or things like substance use, and how that affects the bone remodeling process.
That is all really, really interesting. And it's a lot of bigger words. But I think I just want to step back and talk a little bit about how bones actually work. And then I think that the rest of what you do will make more sense. So I guess let's start with the idea of, so there's two main processes when you think about like bones doing their thing, and that's bone modeling and remodeling. What is the difference between bone modeling and remodeling?
Great question. So I want to preface this with saying that bone is a living record. So it's a really dynamic tissue that's constantly changing over a person's life, and it responds to different factors like injury, or as I said, lifestyle factors like drug or alcohol use. And as a forensic anthropologist, we can retrieve a lot of information about bone from even a small piece, right? Just based on the microstructural organization. So when you talk about bone modeling, this is usually accounting for the growth and sort of mechanical adaptation of the shaft of bones or the diaphyses. So it's allowing bone to grow sort of in length and determines the geometry and position of the bone relative to other tissues. So it's sort of adjusted through the removal of bone from one of the surfaces called the endosteal surface, and then the deposition of bone diametrically on what's called the periosteal surface, or sort of outer surface of bone. And bone is growing through modeling drift. So it's sort of moving through tissue space, and its curvature and robusticity is achieved through this mechanism. So it's primarily happening during the growth process.
Right. And so just to give a little bit of background for listeners who might not realize this, when you're born, you actually have technically a lot more bones than you end up with as an adult, because a lot of your bones are in multiple pieces. So your femur, which is like your thigh bone, is in what, like 3 or 4 pieces when you're born?
So the juvenile skeleton or infant skeleton is composed of a lot more pieces and parts. So like you mentioned, we have these sort of bone caps called epiphyses that are not fused to the diaphysis or the shaft of the bone until different points during growth and development. So this allows it to grow in length and in width, right? And these all fuse at different times. So the last skeletal element to fuse actually is your collarbone, the medial aspect of that.
Yes. And so this actually gets into what part of your research is about, which is that historically, forensic anthropologists, when they are doing age at death estimates, it's a lot easier on people who are not done growing yet, because bones kind of fuse on a very set, regular schedule. Mm-hmm. And so it's pretty easy to, well, it's you can get a better age estimate, like within like a year or two, of someone who is still in the growth process. But once that clavicle finishes fusing and all the bone and all the teeth have finished erupting, after that, people are kind of looking at these like gross, not gross as in ooh, but like big morphological features. And they're looking at degeneration in those features to kind of try and get an age estimate. So for example, if you're looking at the skull, your skull actually has sutures where your skull grows together. So if you look at a baby's head, that soft spot at the top, that's because there's no bone right there. And your bones kind of squish together as you grow older and seal up. So people can look at how apparent the sutures are, but that's like not even 100%. Some people have like really visible sutures even when they're really old. So what you're doing is you're trying to look at like a cellular level of the bone to see if you can figure out how old people are when they die. At least that's one of the many projects you're working on.
Sure, right. Yeah. So you talked a little bit about the conventional sort of gross methods, right? The macroscopic methods for essentially examining the assessment of skeletal features. And you're right in that once our bones are done fusing, we immediately start the process of degenerative change, right? And of course, this is going to be variable depending on the individual's lifestyle, past injuries, things like this. So it's a lot less precise, as you mentioned, than aging a juvenile or subadult skeleton. But it gets tricky to use the macroscopic methods if you have, say, fragmented human bone, or they've been burned or dismembered or otherwise disassociated, right? So the methods that I use in casework and am hoping to further develop are histological aging techniques. So let me sort of circle back to bone remodeling, because we talked about modeling, but we haven't talked about remodeling yet. So bone remodeling essentially is this mechanism of replacing and maintaining mature bone. And so this is what's dominating the bone turnover or remodeling in adult tissue. And essentially this is done through a complex group of cells. So these are known as basic multicellular units, or the BMUs. And these consist of osteoclasts that resorb bone and osteoblasts that form bone. And so your osteoclast will sort of create this tunnel, like resorption space, which is then filled in by a closing cone by those osteoblasts. And so this process is happening throughout your entire life. And our histological techniques are sort of focused on this premise that we have bone turnover occurring at a predictable rate over an individual's life. And so the length of time during which remodeling occurs will be the primary influence on how many sort of microstructural features that remain. So for example, osteons, these are the basic structural units of bone. So we want to sort of figure out how many of those accumulate per unit area of bone.
Yes. I think if I were listening to this and I didn't know anything about bone remodeling, I would want to know, does it happen the same for everyone? Are there things that like trigger it? And is bone remodeling only about like laying down new bone, or how does that work? I think those would be my main questions.
Right. So there are a lot of physiological factors as well as lifestyle factors, as I mentioned, that influence bone turnover. And we don't fully understand all of these processes. So aging certainly has a very strong effect on how bone repairs itself, right? So our bone remodeling process becomes slowed. So you have more resorption from those osteoclasts than you do bone formation. So this leads to increased microfractures, which leads to overall sort of fragility of the bone and more risk of fractures over time. Of course, if you are very active, so even when you're young, but you're running, say, on pavement, all the time, this is going to put a lot of stress on your lumbar spine, on your knees. And so you're going to accumulate more of those microfractures, but your bone will also be sort of triggered to respond to replace with new and healthy bone. And also have called like disuse remodeling. So say I am completely sedentary and I'll have, again, more resorption than formation because I'm not loading those bones.
Right. I think in this case, I would like to imagine you as an astronaut in a zero-G environment.
Yeah.
Because you do not seem like someone who would be inactive to me.
No, I know. I actually, people sort of joke that I'm always, busy doing things like hyperproductive, but it's just, I do that with every aspect of my life, not just work.
I wish y'all could see her. She's like just like a bundle of energy. But anyway, so yeah, this is something that does happen to astronauts, and the way that the way that the bone knows whether or not to put down more bone or to get rid of bone is how much loading force is on the bone, right? So if there's a lot of force on the bone says, oh, well, bones aren't actually saying anything, but for the purpose of this podcast, the bone says, hey, like, there's a lot of force being put on here. We should get a little bit bulkier so that we can withstand the force a little bit better. But at the same time, bones need to maintain the same shape and proportions so that they still work in the rest of your body. So this is actually like a very, very complicated process. Now, the question I think I first had when I was learning about bone remodeling is like, what would be the purpose of losing bone?
Right. So you need to have that older bone, as I mentioned, remodeled out or replaced so you don't have this accumulation of microfractures. So microfractures are going to happen in everyone, right? In healthy people, in young people, in older people, but you need to have this constant replacement of that bone tissue in order to maintain that homeostasis, right?
Right. So like, for example, people who are super, super sedentary and aren't moving around, like maybe they're bedbound or something like that. Bone is a tissue just like any other tissue in the body, and it can be energetically expensive to maintain. So if you're not using, it's kind of a use it or lose it situation. Your body's like, you're not using it. I'm not going to pay in energy to maintain the bone. So resorption.
Right. Yeah, no, that's why it's very important to get into, exercise routines when you're younger. So you're used to sort of, always loading those long bones. And so because there's variability in exercise and diet and hormone intake, all these things, for something like histological age estimation methods, we want to use a bone that's sort of at least consistently loaded in everybody. So a good example of this would be the rib, right? So we all breathe, we all experience respiration. And so that's a consistent biomechanical process we all experience. But again, if you're looking at something like the femur, we're all going to have varying activity levels that will affect the microstructure in that area. So the rib is sort of the primary bone that I examine for not only aging, but other sort of physiological effects as well.
That makes so much sense. I had never really thought about this until I read this review paper that you wrote that was very extensive. It was really, really good. It was really good. I just want to, it was just, I think one of the longer review papers that I've ever read. But as a result, very complete. I had never thought about how much bones would vary on kind of like a microscopic level based on activity, but it totally makes sense. So when you're looking at age-related changes, what are the things that you're looking for on that microscopic level?
Right. So one of the microstructural features that we didn't talk too much about yet is cortical porosity. So for example, in our osteonal systems, right? So we have the basic structural unit of bone, And running within that structural unit is a porous space for blood vessels or nerves. So we're going to have porosity associated with that, as well as the ongoing process of bone remodeling. So we'll have resorptive areas. And imaging cortical porosity is sort of a big push of my current research program, or a big emphasis, because it better helps us to understand age-related changes, disease processes, as well as how bone is adapting to different stressors. And these resorptive areas can be associated with exaggerated age, as I mentioned, or also with sort of a disease or lifestyle effect. And so one of the things I'm really interested in right now is how drug use, specifically opioids, affects the bone remodeling process and subsequent porosity. So I'm interested in that, as well as how it affects the bone cellular network. So are we seeing, a decrease in cells that are involved in bone maintenance and communication, for example, as a result of this type of lifestyle?
Interesting. And do you have any results yet that you're like ready to share?
Sure. So we presented some preliminary research at the American Academy of Forensic Sciences annual meeting last year. So I can talk a little bit about that work. I do have to note that this work is funded by the National Institute of Justice. And so the opinions, findings, and conclusions expressed here are my own.
Thank you. So, Thank you for the disclaimer.
That's a disclaimer. So as I said, this project is basically seeking to describe the effects of opioid use on bone microstructure, so particularly the cortical pores and then the bone cellular network. And I'm looking at the rib. As I mentioned, that's a good site because we have sort of invariable biomechanical strain in that area. So preliminarily, we're seeing that in even young prolonged drug users, we see this decrease in bone cellular density and connectivity of that network. So disruption of that network will increase sort of the fragility and the quality and decrease the quality of that bone. And it's also indicating that we're seeing dysregulation of the cellular activity. And so this does make prolonged opioid use an additional risk factor for things like early onset osteoporosis.
That's really interesting. And one more reason not to do drugs. Just in case you were looking for just that one extra reason, it affects your bone quality.
Exactly. And I was going to say, I can link you to some images that have been already presented if you want to put them on your website or link to them.
Yeah, that would be wonderful.
It's a very visual, 3D bone imaging is very visual.
Yes. So that was actually another thing I was going to ask you is, where are you getting these ribs from that you're looking at? Are they still in the people, or are you using skeletal collections? Like where are you getting your ribs?
Right. So that's a great question. So I've actually been developing my own skeletal collection for histological research since 2017. So this is sort of known as the Andronowski Skeletal Collection for Histological Research, to provide a resource for the study of skeletal microarchitecture variability with advancing age and between the sexes, but it's sort of evolved beyond that. And so I formed relationships with local medical schools, universities, as well as organ procurement, nonprofit organizations, and medical examiners' offices to procure bone for 3D imaging work and histological research. So these are extracted from deceased individuals with the proper ethical permissions in place. And in histology and 3D imaging, sort of a small amount of tissue goes a long way. So we're able to perform a great number of experiments with just a very small, say, 5-inch segment of rib or femur.
Yeah, that's super interesting. I think one of the things that's been touched on by a lot of guests in the show is that a lot of forensic methodologies were developed on historic populations or solely on like a single group of people.
Right.
And so I think it's really interesting that you've like gone out of your way to find a modern group. And what are some of the things that you're like trying to consider when you're well, I mean, part of just for listeners, part of it with any kind of medical research is like whoever you can get, that's awesome, because not enough people are donating their bodies to science. More people should donate their bodies to science. But from the perspective of like actually trying to create metric assessments, what are you thinking about when you're like trying to put together your research group?
Right. So as you mentioned, a lot of the methods that were developed have been primarily on sort of older individuals from European populations. And part of this has to do with just the demographics of people who donate their bodies, right? It's great. Young people are not dying in the same, large numbers, just given aging in general. But the same goes for histological methods. So these were developed using collections that were procured, say, in the 60s from biopsy specimens. Essentially, this is why we actually use the sixth rib, is because of availability of rib samples from a biopsy. And we've sort of built on that over the years. But our aim is to have a diverse collection, so ages, equal sex representation, so males, females, as well as population affinity, right? So have diversity in background as well. And there are limitations because, of course, our donors who sign up, right, at organ donation or for medical school, sort of cadaver research, they tend to be sort of skewed towards, again, older individuals and European individuals. And so there are a number of reasons for this. So there might be opposition for religious reasons or family reasons or personal reasons, right? That the body is not donated to science. So we have our limits, but that's sort of my goal, is to keep collecting samples just over years, right? To sort of build a broad demographic for that.
That's super, super exciting. I think that most of the time when I think of skeletal collections, I think of like the Smithsonian that has had skeletal collections that are like 200 years old. It's not, I don't think about people currently building skeletal collections. So it's just such an exciting, right. You must be such a fun dinner party guest.
Yes, keep in mind that my collection, it's sort of smaller pieces, right? From rib, from femur, from cranial bones. So it's not an entire skeleton. So again, the samples I procure are sort of cosmetically acceptable. So that donor, can go on to have a funeral showing and so on and so forth.
Yeah. So if you're listening to this, I think the two main takeaways we've had so far are don't do drugs and go ahead and donate your sixth rib to the Andronowski lab.
Right. It is ongoing. So there is more information about Asher on my website. And it is open to researchers. You can contact me with a research request and we can discuss using those samples for any of your questions. So that's sort of the goal too, is to have sort of a modern sample that can be used not only by anthropological researchers, but others in the biomedical field.
Yes, that was actually something I wanted to ask you about, is that you're in the biomedical faculty at your university, right? How is that? How is that different from anthropology?
Yeah, absolutely. So as you mentioned, I'm now in a faculty of medicine in the Division of Biomedical Sciences. And so to sort of circle back a little bit, my background was in biology first. So I was a biologist first, right? Then an anthropologist. And so my background and my interests have sort of always been in biomedicine. And I sort of flirted with the idea of being a physician prior to taking an osteology course where I fell in love with bone. But for the type of work that I do and the resources that I need to do it, a sort of STEM department is a much better fit for me. So previous to this, I was in a department of biology. And again, same thing. We have ample lab space, research funds to get the equipment we need to ask the questions that we want to answer, right?
For sure. I think that this can be something that can be like a little bit of a struggle with anthropology. A lot of the time it's considered a social science, and there are parts of it that are definitely much. So if you are a cultural anthropologist, you're a social scientist. It is what it is. But when you're on the biological anthropology side, you kind of are straddling this divide. So you have to kind of figure out where it makes the most sense for you to be able to do the research that you want to do. Another thing that you mentioned to me that I thought was really interesting was that you have a lot of students who are interested in histology, but like don't know how to go about getting involved. And that's something of a passion for you, is helping people get involved. What would your advice be?
Yes, I'm glad that we can discuss this again on the air. So as I mentioned, in anthropology, I do come across a lot of students who are interested, but are sort of nervous to approach questions related to histology because they haven't taken, core science courses like molecular biology or had basic lab training. And so it is more tedious, right? There's a lot of sort of basic skills you need to learn to operate the equipment. But my advice to students is that do a lot of reading, don't give up, right? Reach out to us, reach out to people like me. Since the pandemic has been happening, we've developed training videos of how to operate certain pieces of equipment that are common in a bone histology lab, because preparing undecalcified bone tissue is a lot different than, say, soft tissues that you would be embedding in paraffin and things like this. It's more comparable to the instruments that they use in geology to section rocks, right? Because bone is a hard composite. So I would say that there's just not a lot of instructional literature that exists that sort of walks students through step by step. This is how you prepare a bone section for a histological age estimate, right?
Yeah. So my particular interest in biological anthropology strays more towards endocrinology, which is another thing that within traditional anthropology departments, you don't really find people who have endocrinology labs typically. So when I was in Nebraska, I actually went to a faculty member who studies veterinary endocrinology and asked to work in her lab. So if that's like something that you're interested in, like I don't think that it should be something that stops you, but I think that students sometimes need to be a little bit more creative in looking outside of their particular anthropology department to pursue those scientific interests.
Absolutely. And so my advice, again, for students who are interested in applying for graduate school, look for a professor with research interests who yours align with. Look at them first, reach out to them, see if they're accepting students, check out the program, make sure it's suitable for you, your interests. But I always say, look for the professor first and the program second, right? You might find them in a different department than what you were expecting.
For sure. And I think the other thing to think about is if you can get some lab experience as an undergraduate, I know it can be hard, but if you can, I think it's a good thing to do just because working in the lab is it's different. It's a different skill set. It's like kind of like a different life. So if that's something that you think you might be interested in, I advise that you try it before you go to grad school for it.
Absolutely. I agree. I always tell students apply for, summer research assistant positions, apply through the external granting agencies to get placed at, an alternative Canadian university, and email your professors if you like their course, see if they're accepting summer students or research students, because often we are. And it's great to sort of figure out what works for you, and something that you think might work for you ends up not being, your best fit. So I think getting diverse experience is great. Like I worked in, insect behavioral ecology labs, microbiology labs. I worked in medical examiners' offices, all these things as undergraduate, and I learned from all of them, right? Different skills.
Yeah, for sure. I think the other thing that I think about too, though, is that this is something that I talked about with Dr. Tallman, who incidentally you are friends with, which is that sometimes it's hard because I'm sitting here saying like, you should try to do these things and like, it'll help increase your chances of getting into grad school, but it'll also like help you know what you actually want to do. But at the same time, I think that it can be hard to hear these things if you don't have access to them and you're like, so now what? You know what I mean? Because not everyone has the ability to access these things in undergrad. I don't think that should discourage you from applying though. What do you think?
Of course. Yeah, as I said, I think a lot of students are hesitant to reach out to faculty members, but the worst that happens is they say they're not taking students or they don't respond, but it's still worth a shot, right? I say that too about grant applications. If you don't apply, you're definitely not getting the grant, right? And there's, now with working primarily remotely in a lot of places, there's been more opportunities to get involved doing work for professors in different locations, right? So I have a student group, for example, that I manage called the Biological Anthropology and Human Anatomy Student Club. And that started at Akron, but since we've gone virtual, we have members, from New York City, from Argentina, from all these places. And it's really grown sort of the diversity of our group and gotten people involved who otherwise wouldn't have been able to.
Yeah, I think I'm not going to say silver lining. I hate people who are like, that's one of the silver linings of the pandemic. Because it's like, honestly, this is a terrible time. But I think that one of the things that is coming out of this that is good for a lot of people is showing that online work is possible and it can make work a lot more inclusive.
Absolutely. And, with my research, a lot of our work involves working with large data sets and image processing. And we have right now I have about 11 terabytes of data I'm working to process. And so this can be done from home. We can remote into our workstations and work on that from home. So that's been very great for us when we were shut down and couldn't do in-lab experiments last year.
So it's been a while since I've harped on this, but math's cool, huh?
Absolutely. I always tell my students, don't be afraid of the hard sciences. Take your biology, your chemistry, your math, your physics. And again, my advice to students who want to go into biological anthropology or forensic anthropology is to become a scientist first, forensic scientist second, right? So do a degree in those core sciences versus a degree in forensic science. And that's my advice.
I think that's really good advice. I think that's really, really good advice.
Because as you said, like if you get into, say, a graduate program and then you decide it's not for you in anthropology, if you have sort of a broader background to fall back on, with a biology degree, you can have a lot of opportunities in various labs or even sort of forensic type positions working with police departments, things like that.
For sure. I totally agree with that. Is there anything else that you would like to talk about that you haven't gotten to talk about yet?
Let me have a think. Another sort of tidbit of information, I guess, for students that I'd want to add is if you can take a gross anatomy course or anatomy and physiology course during undergraduate or during your graduate program, that will be a very big asset to you on the job market. I do tell my students that there are many different types of universities and programs that require anatomists, right? Pharmacy schools, dental schools, medical schools, nursing colleges, all of these things require anatomists. So I am teaching gross anatomy to medical students at MUN. And I love being in the lab. I love dissection. And so this has really opened a lot of doors for me. And so I would definitely recommend students seek out those opportunities. Also for TA-ships, if there's a TA-ship, even if it's outside of your department. Sean and I both TA'd in the biology department during our PhD programs and taught students gross anatomy. And now we're both in anatomy departments.
Yes. As an undergraduate, I think that was actually the most valuable class I ever took, was my human anatomy class with a human cadaver lab. And if you can, because a lot of the times, this is pro tips here, a lot of the times there's like a limited number of cadavers to dissect. See if you can get into the first lab section, because then you actually get to do the dissection. Some labs aren't like this. Some labs only let the TAs do the dissection. But if you can, as an undergraduate, if you get into the first lab section, that is ideal. I think you learn a lot from the dissection itself.
Absolutely. And I had an undergraduate anatomy program at Akron, and my students assisted with dissection. I trained graduate students, undergraduates, and started that program there because it's so important to me that students get that hands-on experience and learn the 3D spatial relationships between structures in the real body. So that is very important to me. And we actually have a brand new cadaver lab that's set to open in the next couple of months here at MUN. And I'm very excited to sort of spearhead the development of that and incorporate cadavers back into our program because there's been a bit of a hiatus from renovations and pandemic.
Yeah, of course, pandemic. Okay, well, thank you very much. We have reached the portion of the interview which I actually don't think I warned you about, so apologies. But I always ask my guests 3 questions about what they're reading or what they're consuming. So my first question for you is, if someone wants to learn more about your particular area of research, where's a good place for them to start?
Okay, so there is a great edited volume that I'm going to grab off my shelf so I'll give you the exact title of it.
Okay.
Written by my mentor, Christian Crowder, and Sam Stout, who's at Ohio State University. So it's called Bone Histology: An Anthropological Perspective. And this volume has a lot of really informative chapters in general about bone histology, basic bone biology, like we talked about bone remodeling, bone modeling, growth and development mechanisms, how to differentiate human from non-human bone using histology, which we didn't actually get into today, histological age estimation, as well as technological developments in the field. So 3D imaging, like iApply, for example, there's a chapter on that from another one of my colleagues, Dr. David Cooper, who's here in Canada as well at the University of Saskatchewan. So this is a fantastic resource, Bone Histology: An Anthropological Perspective.
That is like the most complete and like ready answer I think I've ever had to this question on the podcast.
I direct this volume to all of my students who are starting out. Sometimes your library can get some of the chapters through interlibrary loan and then send you the PDF of the specific chapters. I sort of have a bone biology library in our shared team drive for my students. And so we don't have obviously the PDF in there, but I recommend that they get this volume number 1.
Thank you. I really appreciate that. A question just occurred to me. I don't know if you know the answer to this, but have you ever been on an archaeological dig site and an archaeologist is like, I wonder if this is bone, and then they lick it? Does that actually work?
So I've heard of this happening. I remember even my undergraduate advisor telling me that this was something that they would do on archaeological excavations, but I have never done that, and I would not recommend it.
Listeners, just in case you've never, archaeologists have this idea that you can distinguish bone from like other substances by licking it. I don't know what licking it is supposed to do, but probably just don't lick anything that you might think might be biological in origin. Don't lick anything that comes out of the ground, actually.
No, yeah, and that's the thing, right? Like with bone, for example, we don't know what the history was of that individual. There could be disease, even in ancient bone, you can extract tuberculosis DNA and things. So
okay, so 3 messages from this podcast. Don't do drugs. What was the second one? Donate your body to science, and don't lick bones.
and study biology.
Oh, and study biology and math. We've got so many positive messages on this show. My second question is, what are you reading for work right now?
For work. So we're always reading new articles, right? So every week I have what I call having a think time where, I sit in a comfortable chair and I read a selection of new papers that have come out. I'm actually sort of taking a different trajectory with a project I'm working on since I got to Newfoundland. And so it's actually about a cardiac condition that's called arrhythmogenic right ventricular cardiomyopathy. There are variants that can be highly lethal, and this is very prominent in this population. And so we're looking to sort of classify different histological findings based on sections of the heart and automate if we're seeing fat or fibrosis replacement in the myocardium, which is the muscular tissue of the heart. So I've been actually reading a lot about hearts and still bone biology, but that's sort of a different trajectory.
That's really interesting. I've never thought about that before. Third question, final question. Congratulations, you've reached the end of the interview. Is what are you consuming for fun right now?
So as I said, living in Canada again, I'm living in Atlantic Canada in a very beautiful part of it. And so Newfoundland has the famous East Coast Trail system. And so I've been doing a lot of hiking. The weather has actually been abnormally warm. We do not have a lot of snow and have not had a lot of snow this winter, which again is not the norm. And so I've been just going to as many hiking trails as possible, taking in the cliff views, the ocean views, hopefully see some icebergs in the coming months. So that's sort of been my personal goal, is explore my new island.
That's beautiful. That's so nice.
It is. It's a beautiful place.
I'm really jealous, actually. I feel like I never really thought about Canada growing up, and honestly, I've never been, which is crazy to me because I have spent so much of my life traveling. Both my parents are immigrants, so pretty much every summer I was sent out of the country to visit one relative or another, and I've just never made it to Canada, and I don't understand why.
Definitely. I recommend one of the coasts. So I've lived in Vancouver as well. So West Coast, beautiful, lots to do. East Coast, beautiful, lots to do. Sort of so many different cultures right across Canada. Like I'm from the Toronto area, which that's great too. Big city feel, lots to eat, lots to drink, lots to explore, lots of museums, art, that kind of thing. But definitely I recommend one of the coasts, especially if you like outdoor activities.
Yeah, I definitely do. That's one of the best parts about living in San Francisco, to be honest. Okay, this is turned into an entirely different discussion. Thank you so much for coming on the show. I loved having you, and I hope you have a wonderful day.
Great. Thank you so much, Gabby. I appreciate that.
Thanks for joining me for this episode of the Anthrobiology Podcast. If you have questions or comments, you can reach me at gabby.lapera@anthrobiology.com. That's spelled GABY.LAPERA@anthrobiology.com. This show was produced and edited by me. Music courtesy of Chad Crouch, aka Poddington Bear at Sound of Picture.